Okay, this is Applied Anatomy and Physiology Lesson 5 on transport of oxygen and the Bohr shift. Hopefully by the end of the lesson you'll be able to describe how oxygen is transported around the body and be able to describe and explain something known as the oxyhemoglobin dissociation curve, leading to knowing about the Bohr shift. Okay, so firstly, when you breathe in oxygen, most of it is actually combined in the blood with hemoglobin to form oxyhemoglobin, 97%. 3% of that doesn't actually combine with hemoglobin and just dissolves into the plasma and is carried around your body in the blood. In the tissues, such as your muscle tissue, oxygen is released from oxyhemoglobin to the tissues, known as oxyhemoglobin dissociation, and in the muscle it is taken up and stored by something called myoglobin. Now, myoglobin has a large affinity for oxygen, higher than hemoglobin. So when the oxygen is carried by the hemoglobin to the muscles in the blood, when the muscles are exercising in particular, the hemoglobin lets go of the oxygen and the myoglobin takes on board the oxygen within the muscles and takes it and stores it where the mitochondria is using it to produce energy. Now, what you need to do is we need to understand how that actually happens. So this is known as oxygen dissociation. So if you watch the video and play it back a few times and make sure you're happy with what the video is showing, feel free to draw out the graphs as they are in the video.
Hemoglobin and oxygen dissociation curves. Hemoglobin is a large protein made up of 4 polypeptide chains, so it's described as having a quaternary structure. Each polypeptide chain has a heme group containing iron. Each polypeptide chain binds to one oxygen molecule, so one hemoglobin protein can carry a total of 4 oxygen molecules at once. If there were to be a fault in the hemoglobin's DNA, then a wrong amino acid would be made, meaning an incorrect polypeptide would be made, and so oxygen would not be able to bind. High affinity is when oxygen can bind easily to the hemoglobin, but it struggles to be released, and low affinity is the opposite. Oxygen struggles to bind, but it can be easily released. The role of hemoglobin is to carry oxygen around the body. Associated gas exchange surfaces load oxygen, and dissociate at the tissues, unload oxygen. Hemoglobin can be found in earthworms, starfish, insects, plants, and even some bacteria. An oxygen dissociation curve shows how saturated the hemoglobin molecule is with oxygen at any given partial pressure. And how saturated the hemoglobin molecule is depends on whether it has a high or low affinity. It's very difficult for the first oxygen molecule to bind to hemoglobin, but once it does, the protein undergoes a conformational change in shape, which makes it easier for the next 2 O2 molecules to bind to it. But then, as the hemoglobin becomes more saturated, it is harder for the oxygen to bind to it. Steep sections of the dissociation curve are where O2 binding is easy, since hemoglobin has a high affinity, but the shallower sections are where O2 binding is difficult. Sometimes an oxygen curve will shift to the left or to the right. This is linked to the affinity. If the curve shifts to the left, hemoglobin has a high affinity for oxygen. If it shifts to the right, hemoglobin has a low affinity for oxygen. The Bohr effect is something else you need to remember. The greater the concentration of CO2, the more readily hemoglobin releases oxygen. When cells respire, they produce carbon dioxide, which raises the partial pressure of CO2. Since there's a greater concentration of CO2 in respiring tissues, the rate of unloading by hemoglobin increases. Therefore, the curve shifts to the right, because hemoglobin now has a low affinity and can unload oxygen easily. So, to summarise, high affinity, oxygen binds easily and is difficult to release. Dissociation curve shifts to the left. Low affinity, oxygen is difficult to bind but easy to release. Dissociation curve shifts to the right. Hemoglobin has a low affinity for oxygen when it meets CO2. And that's it. Thanks for watching.
Okay, so use this next slide just to summarise what we've looked at. So we need to know what an oxyhemoglobin dissociation curve is, and we need to understand firstly what it's showing, and then to understand that during exercise the S-shaped curve shifts to the right. And the reason it shifts to the right is because there's more oxygen dissociating or leaving the hemoglobin when the actual blood gets to the muscles during exercise. We need to understand the three causes of this Bohr shift. And the three causes when we're exercising are an increase in blood temperature, an increase in carbon dioxide, and a decrease in pH. So you might need to watch this video a few times to get your notes and get your ideas, and write down any questions that you may have to ask in class.